All right, well, we've covered the first half of chapter 5, and we learned about cellular respiration. We used as an example of oxidation of glucose for producing energy. We talked first about glycolysis, a series of reactions that breaks down glucose into pyruvic acid, liberating some high-energy electrons and producing some ATP. Then we took the pyruvic acid and decarboxylated it, producing acetyl-CoA, which we entered down into the Krebs cycle, a circular set of chemical reactions that also produce some ATP and liberates some high-energy electrons, produces CO2 as well. We take all the high-energy electrons from glycolysis and Krebs cycle, enter them into the electron transport system, and then as the high-energy electrons are passed by redox reactions from protein to protein in the bacterial membrane or in the inner mitochondrial membrane, pump hydrogen ions across the membrane, and as they diffuse back down through, they generate ATP through this protein complex called ATP synthase. Well, at the very end of that electron transport chain, we said that in one instance, in aerobic respiration, oxygen becomes the final electron acceptor. You see the electrons being passed along, and finally they're taken away by oxygen and wind up producing water. That way there's a space in there for additional electrons to keep coming through this chain. Otherwise the whole thing would back up and stop. So we got to take away the electrons. Anyway, aerobic respiration, the electrons are taken away by oxygen. Bacteria, again, and microorganisms are very diverse. Some use nitrate ion to catch those electrons, producing nitrite or elemental nitrogen and water, or sulfate ion to ultimately produce hydrogen sulfide gas, or carbonate ion, which ultimately produces, as an electron acceptor, produces methane gas and water in each case. So pretty cool. Some very diverse organisms indeed, and some interesting products. Hydrogen sulfide has that kind of rotten egg smell. Methane makes a fine fuel. Carbohydrate catabolism, just a little summary look at some things. Glycolysis takes place in the cytoplasm of both a eukaryote and prokaryote. Of course, prokaryotes, everything takes place in the cytoplasm, pretty much. The intermediate step, the decarboxylation of pyruvic acid, cytoplasm of eukaryote and prokaryote. But then the Krebs cycle chemicals, all the Krebs cycle enzymes and chemicals and intermediates are inside the mitochondria, the so-called mitochondrial matrix in eukaryotes, but in the cytoplasm of prokaryotes. And where's the electron transport chain? Inner mitochondrial membrane and in the plasma membrane of bacteria, as we mentioned in the last session also. Just tallying up the energy production by cellular respiration. Oh, there I go again. In the glycolysis, we saw there's a net 2 ATP produced. Krebs cycle, 2 ATP are produced. So by the time we finish through those, we have 4 ATP already. And then we produce a whole bunch of NADHs, 2 during glycolysis, 2 during that decarboxylation to produce pyruvic acid, 6 NADHs produced during Krebs cycle for a total of 10, and FADHs 2 produced during Krebs cycle. Each one of these 3 NADHs, I'm sorry, let me start over. Each NADH will produce an equivalent of 3 ATPs in chemiosmosis, and each FAD will produce an equivalent of approximately 2 ATPs. So now if we feed all those things through, we therefore wind up with 4 ATPs initially produced by substrate level phosphorylation and 34 ATPs produced from those electrons in the electron transport chain in chemiosmosis. So 38 ATPs are possible to produce during aerobic or during cellular respiration. In eukaryotes, we say that there's only 36 ATPs produced. There's some slight details that we aren't going to worry about. Generally 36 to 38 ATPs produced. Some tallies suggests only 34 are produced in, say, muscle cells of eukaryotes, but that's not really an issue. All right, let's talk about our next topic, fermentation. It's the last part of our sort of metabolic diagram we've been following along with all the first half of chapter 5, the fermentation process. When we talk about food spoilage, we're generally talking about fermentation going on that very much changes the properties and the flavor and smell of food. Um, there's a whole bunch of different fermentation products that we know about. Alcohol is produced intentionally as a fermentation product in certain beverages. Um, acidic dairy products like yogurt are fermentation products. Let's look at a list of some possible examples of fermentation in just a second. So what is it? After glycolysis, if we're not going to send the pyruvic acid down into Krebs cycle, we've got to get the NADH that was produced during glycolysis back in the form of NAD to allow glycolysis to continue. So again, if we're not going to use Krebs cycle and electron transport chain to get rid of the electrons, we've got to do something else with them, and that something else is going to be fermentation. So here we are in this right-hand arm of this diagram, which is the only place we haven't really approached yet. Just talked about in detail. So in this case, we're talking about a situation where we're not going to send the pyruvic acid, we're not going to decarboxylate it and send it down into Krebs cycle as acetyl-CoA. Instead, we're going to send those electrons that were on the NADH right back onto pyruvate to produce lactic acid or possibly some other end products resulting from that. So that's fermentation, sending those electrons and hydrogens back onto pyruvic acid. And look what's happening. The NADH is being restored back into the form of NAD, which can then come around and enter and do some more glycolysis and produce fresh NADHs. So you can't do glycolysis without that coenzyme NAD present. So that's the deal. That's why fermentation takes place, to restore NAD for further glycolysis reactions. So here's a little even more expanded out um, explanation. Glycolysis, take the pyruvic acids. We're not going to do Krebs cycle and electron transport. What are we going to do? We're going to take the electrons and hydrogens and stick them right back onto pyruvic acid, restoring the NADHs and producing fermentation products. So that's the fermentation reaction or step. We can either produce just lactic acid, so-called homolactic fermentation, right? Your muscle cells do that right in eukaryotes in your own body, but many microorganisms can produce lactic acid, or they can produce lactic acid plus other fermentation products beyond that, sort of heterolactic fermentation, and we'll look at some other compounds that are going to be produced instead of just lactic acid. Here's an example of alcohol production instead of any of these lactic acid involving steps. We're going to take the pyruvate and produce 2-acetaldehyde and convert those into ethanol. Over here we're producing lactic acid and possibly go on to produce some other fermentation products from it. Many microorganisms, as you can read here, are identified on the basis of which products they can ferment. In other words, we've been talking about fermenting glucose, taking glucose, running it through glycolysis, and then producing some fermentation product like lactic acid or ethanol. But some microorganisms can ferment other sugars. And so those unique properties will help us identify specific microorganisms. So here's a picture of a microorganism that was set up in a reaction where it might have fermented a particular sugar. Let's say lactose was present in there, but it did not ferment it, and this pH indicator remained orange. And then over here we have a bacterium that very avidly fermented this particular, in this case maybe it was lactose, and the pH indicator is becoming acidic as we produce some kind of acidic fermentation products. The media is turning yellow. Lets us know, yes, that microorganism can ferment whatever sugar that we put in there for it to utilize. And now we know something about the bacteria, what kind it is. Also, here's a little a Durham tube, it's called. This is an upside down tiny glass test tube, which was full of media at the start. And some microorganisms produce some gas while they're fermenting, and that can be, again, a telltale sign to help us identify a particular microorganism species. And yep, we see a gas bubble that's formed in there during the fermentation. That gives us, again, a little clue about what organism is present that we might have isolated from the stool of a sick person. So a bunch of different fermentation products. Ethanol in beer and wine production. Also, ethanol is sometimes used as a fuel. The ethanol got popularized as a fuel additive for gasoline for automobiles and trucks. The idea was that we could reduce our dependence on fossil fuels by adding in some ethanol that was produced by fermentation of sugar from corn, say. And that was the idea. It didn't turn out to be a huge breakthrough in terms of energy efficiency or anything like that. So it's not as popular as it was, but it's still, that's every time we use ethanol, we use a little bit less oil that comes largely from the Middle East and also from our own limited resources, finite resources in our continent. Acetic acid, vinegar, right? You go to the store and buy yourself a nice bottle of vinegar for various uses. How is it produced? Fermentation reactions. Lactic acid, certain cheeses and yogurt, sauerkraut, summer sausage, all these familiar food products are the fermentation products containing lactic acid. Swiss cheese, propionic acid and carbon dioxide are producing the large hollow spaces in the Swiss cheese. And a whole bunch of different products. Citric acid, flavoring. Methane is an important fuel that's produced by some microorganisms. That's another way of harnessing waste products from farming animals, from domesticated animals, for example. We can produce some methane from the microbes in their feces. Missionaries have been doing it for generations. Sorbos to produce vitamin C. So some really interesting, diverse fermentation products that different microorganisms produce. Some of them are listed right over here if you're interested in checking on that. Do not worry about memorizing anything from this table. It's just for interest's sake that fermentation is an important industrial and agricultural way of harnessing the properties of microorganisms for our benefit. How about lipid catabolism? Lipids, typically we're talking about fat, which is triglycerides, and a triglyceride is made up of a glycerol backbone of 3 carbon alcohol, so it's just carbon, carbon, carbon with 3 OHs on it, and when it's in the form of fat, it has 3 long-chain fatty acids attached to each one of those alcohols, and so it's a triglyceride. When we metabolize it, we break off those fatty acids and release the glycerol, which can be used to pass through the glycolysis, some of the catabolism pathways of carbohydrates, and we can produce pyruvic acid and get energy from it, or it can actually be converted to glucose. Combine 2 of those 3 carbon glycerols to make a 6 carbon sugar. Over here, fatty acids can be broken down 2 carbons at a time into acetyl-CoA. Remember, the acetyl group of acetyl-CoA is a 2 carbon moiety. Typically we start out with in carbohydrate oxidation with pyruvic acid, which is 3 carbon. We decarboxylate, we wind up with the 2 carbons, and then we feed that into Krebs cycle down here. While in the case of fatty acids, a series of enzymes in the cytoplasm will break down by beta oxidation, it's called, fatty acids into acetyl-CoA, lots of it, and that can enter the Krebs cycle and produce us some energy by chemiosmosis. Pretty cool. But remember, beta oxidation, or acetyl-CoA, is the entry point for oxidation of fatty acids, so we cannot utilize glycolysis in that scenario. We can only enter in, we have to have Krebs cycle and electron transport working to utilize those fatty acids. Right, with carbohydrates, if we want to, we can just do glycolysis and keep producing 2 ATPs for glucose and doing fermentation, just getting lots of ATP that way. Fatty acids, nope, got to go down into Krebs cycle electron transport to get any ATPs out of the deal. Protein catabolism. Proteins are broken down into their building blocks, amino acids. Amino acids, there's a whole bunch of different ones that have different numbers of carbons and so forth, different side chains, different organic chemical groups on there. So that's why there's all these different arrows. What typically happens during catabolism of amino acids, we deaminate, take the nitrogen off of there, and then we wind up with a keto acid, it's called. It has no more longer the nitrogen, and the keto acid, depending on what its identity is, can be fed into different places along this catabolic and energy producing pathway. There's some of the Krebs cycle intermediates are just the same as a deaminated amino acid, so they can feed right in there and so forth. So that's how they can become utilized for energy. They just pop right in here, and we work our way down into the electron transport chain and chemiosmosis. I would point out that in some organisms can utilize proteins for energy, some sources, some can't. So again, that's going to be another way to help us identify different organisms. So we have lipids coming in here as fatty acids being broken down to acetyl-CoA. We have glycerol coming into the glycolysis pathway, can be produced into pyruvic acid and into energy production. Now we're adding proteins in there, broken down to amino acids, deaminated, and then entering into the pathways that way. Here's a experiment to demonstrate or distinguish between microorganisms that cannot or can utilize amino acids for energy. And so in this case, there's a pH indicator that has remained neutral or more acidic, and it's an organism that did not use the amino acids for energy. In this case, the amino acids were being deaminated and producing ammonia, which is an alkaline pH, which made the media turn bright pink. I don't know if we have that broken down to that level of detail. We do not. Oh yeah, the amine may be converted to urea or ammonia, in which case we'll see ammonia might be, or urea might be broken down into CO2 and ammonia, and we'll see this pink color. So that's another way of identifying microorganisms that we already know can break down amino acids for energy that way. Here's another test that helps us identify a microorganism. Is hydrogen sulfide being produced? Remember earlier we saw that hydrogen sulfide might be a product if we use sulfur as an electron acceptor at the end of the electron transport chain. And so here we have stabbed some microbes down into this agar medium. And in the case that hydrogen sulfide was produced, this particular media turns black. So that lets us know, yes, there was hydrogen sulfide being produced. And again, that helps us identify, distinguish between microorganisms that cannot or can produce hydrogen sulfide. Yet another test that can be used to distinguish between different microorganisms. Some microorganisms can take urea, that's a product of sometimes produced by deamination of amino acids, break it down into ammonia and CO2, and if that's possible, that would indicate the presence of an enzyme urease. And so if this microorganism can do that, it'll produce a pink color because again ammonia will make a basic pH and the pH indicator will turn pink. In this case, the microorganism did not have urease and could not break down urea in the medium into ammonia and CO2. So just multiple ways we can test and identify. So here's a little flow chart, a series of tests that might be used to identify a particular organism, Mycobacterium tuberculosis, from an acid-fast bacterium of an unknown identity. We could do some tests. Is it slow growing? Because we know that Mycobacterium tuberculosis and leprae and so forth are slow growing. We'll do a urease test if it's positive. Then we might do a nitrate reductase test and see if it's able to utilize that nitrate. And then if so, then we know the only possible organism that would be positive in all those that series of tests is Mycobacterium tuberculosis. And you'll be doing that exact same thing. It's a very fun thing to do in the laboratory for those of you taking micro lab, where you'll be identifying an unknown bacterium by doing a series of logical steps or tests, biochemical tests and various types of tests. And eventually you will specifically identify the organism that you've been given as your unknown in the microbiology lab. Very fun to do. Photosynthesis. Let's take a brief look at photosynthesis, a very interesting and important topic when it comes to microorganisms and plants. For our interest, we're thinking about microorganisms here. Photo using light energy for something, in this case to produce ATP. That's called the light-dependent reaction. So photosynthesis is two parts. We're going to use the energy from light to produce ATP and possibly also capture some high-energy electrons in the form of NADPH. And then we're going to do a synthesis step. We're going to capture some CO2 from the atmosphere and fix it, meaning grab it and assemble it together to produce an organic compound of more complexity, some sugars. So the the light-dependent reactions are the ones that need the chlorophyll and capture the light energy and excite some electrons. Dark reactions are so-called are the ones in which we're going to actually produce a synthetic product, the sugars, in the so-called Calvin-Benson cycle. We won't say much more about these light-independent reactions anymore here because I'm not sure why, but we're going to stick with just talking about the energy-dependent reactions, the light reactions. We said that chromatophores are a possible location in some microorganisms where all these photosynthetic reactions are taking place. Little invaginations of the cell membrane underneath the cell wall of these guys, where that's happening. Photosynthesis can be oxygenic, meaning it's going to produce some oxygens. If we use water as a source of electrons for the photosynthetic process, we're going to wind up producing oxygen as a product. Isn't that interesting? It's the exact reverse of the electron transport chain and using oxygen as an electron acceptor to produce water during energy production. Now we're doing everything in reverse. We're going to produce energy using light photons and possibly produce some oxygen, but not always, because in the case of some organisms can use hydrogen sulfide, or actually use hydrogen sulfide as the energy or as the electron source and produce sulfur in the end, and not oxygen at the end of the process. That may be a little bit unclear. It'll be a lot easier to look at it in subsequent diagrams and see what I mean by that. So here's our photosystem 1, it's called. In this case, we have chlorophyll accepting some or capturing some light energy. Some photons are going to hit the chlorophyll and boost up some electrons to a higher energy. Then we're going to pass them through a series of redox reactions from a carrier to carrier in the electron transport chain, and then back to the chlorophyll. And as the energy is given up by those electrons, we're going to harness that to produce ATP, just like we did in the electron transport chain of their mitochondria and bacterial cell membrane. In this case, though, we're using photons for an energy source instead of using electrons from oxidation of glucose. Here's photosystem 2 and photosystem 1 that are going to be used together for a different endpoint. This is called cyclical photophosphorylation because we can just keep on sending these electrons around and around, exciting them, letting them trickle back down, exciting them, letting them trickle back down, and producing ATP indefinitely as long as there's a light source. In this case, we're going to get some electrons from someplace else. Here's some electrons coming from water. We have the right enzymes to do this trick. The electrons will be added to the chlorophyll, excited by the light photons, and here they are boosted up to higher energy, coming to photosystem 1, which they'll be excited by electrons boosted up, and they'll trickle down through the electron transport chain. But in this case, we're not going to cycle them back to the chlorophyll. They're going to be ultimately used to produce NADPH. This coenzyme now is going to capture those electrons in the form of NADPH to use. You know what's going to happen to that? It's going to go right to the dark reactions, and that's going to drive the production of sugar in the dark reactions, Calvin-Benson cycle. So it's a different purpose. Now we want to synthesize something. We don't just want to get ATP energy out. So we'll then send the ATPs and the NADPH down into the dark reactions to produce sugar. So again, we could use water as an electron source to load up this chlorophyll, leaving oxygen behind, or we might use hydrogen sulfide down here to give up the electrons and produce some sulfur down here. But so different, a variety of capabilities of different organisms to do this kind of non-cyclic photophosphorylation. We don't need to worry too much about this. This is a table showing us some different possibilities for different microorganisms that will use um, sulfur and sulfur compounds and hydrogen gas um, versus sulfur and sulfur compounds and hydrogen gas. We're not going to worry. I don't want to go through this whole table with you. If you're interested, you can certainly look in there and check it out. We have enough details in this chapter, I think you'll agree. All right, so just putting some things together. We're going to get ready to classify microorganisms in terms of their energy source and their carbon source for everything that they do. And to start out with, we'll say, okay, what's the energy source for different types of microorganisms? That you see they're going to be photosynthesis, photons of light exciting electrons, or it's going to be organic molecules. And we're going to oxidize those organic molecules, or in this case, inorganic sulfur, ammonia, some chemical source of energy, high energy electrons that can then be used to produce ATPs. And so here we have photosynthesis, here we have chemicals as our energy source, our electron sources. When I say energy, that's really what I mean by is high energy electrons. Electron carriers, either NADPH or NADA and FAD. Either one can capture electrons and transport it to the next stage of an energy production system. And we might use oxygen as an electron acceptor at the end and produce water. We might use nitrate or sulfate or carbonate, as we saw in that first table of this part 2 section, to capture electrons and allow energy to be produced through the electron transport chain. Or you might not do electron transport. You might capture the electrons on an organic compound like pyruvic acid and do fermentation. So now we kind of see how we're putting all the things together. It's different sources of electrons, energy, and different outcomes, different locations where we get rid of those electrons in the end. So here we go. We're going to classify all of our microorganisms into 4 categories. They're either chemoheterotrophs, chemoautotrophs, photoheterotrophs, or photoautotrophs. The first half of each one of these compound words refers to the source of energy. Was it photosynthesis or was it chemicals? So there you see the photo versus chemo. That's the energy source. And then the second part is, where do the carbons come from for building stuff? And the carbon source could be CO2. If photosynthesis is happening and you have the ability to fix CO2, you have the dark reactions, the Kelvin-Benson cycle, for example. We can capture CO2 and make sugar out of that. That's the source of carbon. So now that's called that's an autotroph that can capture CO2 and make some organic compounds out of it, that carbon and oxygen. Whereas in many organisms that's not possible. They have to have organic compounds coming in from some other source. Again, our bodies, all of our cells need glucose or fatty acids that we take in from outside the body. Fungi take in all of their organic compounds from outside the body. That's what their carbon source is, so they're heterotrophs. So chemoheterotrophs have their energy comes from chemicals, and they get their carbon from other chemicals that they bring in. They don't make it from scratch. Chemoautotrophs get their energy from chemicals, but they can fix CO2 and produce carbon compounds that way and produce sugar. Photoheterotrophs are ones that can use light photons to make energy for their energy source, but they use chemicals as a source of carbon. They don't have this set of reactions here where they can, the result of the reactions that aren't shown there, the Kelvin-Benson cycle, they can't do the dark reactions and produce organic compounds, whereas photoautotrophs, a typical plant, can use photons for energy and can fix CO2 and produce carbon compounds. So now you see how you define this. Always think the first half of the word is the energy source. The second half of the word is the carbon source. It's either auto, meaning CO2 fixing, or it's hetero, meaning you have to get the stuff from outside. And some examples of microorganisms. So that's a pretty cool thing. You definitely should be able to explain what those 4 terms mean in terms of energy source and carbon source. You don't need to be able to name all these organisms down at the bottom, but I do want you to know what those mean, those terms. So here's an even easier than that diagram probably way to look at it. Photoautotroph, source of energy, light. photo. What about its carbon source? Autotroph. It can fix CO2 and produce its carbon compounds. Photoheterotroph. Energy source, light. Carbon source, no, it cannot fix CO2. It needs to bring in its organic compounds, meaning its carbon source from outside. Chemoautotroph, it uses chemicals for energy, and in this case, it can fix CO2 using chemicals for energy, chemoheterotroph, and brings in its carbon from outside from organic compounds. Yeah, this might be a good thing to just practice with this chart, that you can define each of these four classifications of microorganisms. Microorganisms can also go on in certain cases to produce polysaccharides. So that just means polymerizing together a bunch of sugar molecules into a larger molecule. Glycogen is a storage form of glucose. It's a polymer of glucose. So some microorganisms can produce glycogen. And they do that by starting with ADP glucose, and they can assemble a bunch of those into glycogen. Some microbes or animal cells use UDP glucose. Don't even worry about that. UTP versus ATP. Uracil is just a unique nucleotide sugar-making starting material. We're not going to go into that at all. Peptidoglycan is the bacterial cell wall. You need a different starting material to produce the polysaccharides in that. So we're not going to, I don't want to spend any more time than that on that. As far as details, you do not need to memorize these intermediates that are used to, we start with glycolysis and we can produce these ADP glucose, UDP glucose, UDP N-acetyl glucose. We don't need to worry about any of that. Just so you know that there are pathways, specific pathways to produce polysaccharides in microorganisms. Biosynthesis of lipids. Lipids are important in various processes and pathways. We can start out with glycolysis, take a glucose molecule and break it into that 3-carbon form, and that can be converted into glycerol. That's that 3-carbon backbone for a triglyceride or a diglyceride, a phospholipid. And then we can take acetyl-CoA, right? Pyruvic acid can be decarboxylated to produce acetyl-CoA, familiar reaction. In this case, we said that fatty acids enter the energy production scheme here at acetyl-CoA. Well, some microorganisms have enzymes that can reverse that process and produce fatty acids from acetyl-CoA, snapping together 2 carbons at a time to produce lengthening chains, 16 to 18 carbon long chains, fatty acids, and then make triglycerides. Amino acids can be synthesized by reversing the exact reactions we saw before. The pentose phosphate pathway, that 5-carbon sugar pathway, can produce some intermediates that can then be used. They can be aminated. We can add a nitrogen group on there and produce amino acids, or we can take intermediates from Krebs cycle, various places along the Krebs cycle, and aminate those and produce amino acids. Entner-Duderoff pathway, that alternative to glycolysis, produces some other intermediates that again can be used to produce amino acids. Now we can get the full complement of amino acids. Sometimes, though, the amino acids aren't produced directly from these 3 processes. They're produced, sometimes we can only produce maybe 1 out of a pair of amino acids, but then we can produce the other by transferring the amine group, the nitrogen group, from one amino acid to the other. So here we see, starting out with glutamic acid and oxaloacetic acid. That's the first chemical of Krebs cycle. And a transamination is a reaction in which we take the amine group and transfer it from the glutamic acid, in this case, to the oxaloacetic acid, to produce aspartic acid. This is a different amino acid over here. We're left with alpha-ketoglutarate. So transamination is swapping nitrogens among compounds to produce a little bit more variety of amino acids, just so you know what a transamination is. Purines and pyrimidines are the nucleotide bases that are used for making DNA and RNA. Not going to touch it at all. I just want you to realize that microorganisms have to, in order to make their DNA and RNA, they have to have a way to either take in nucleotide bases from outside, like we do in our bodies, or we can also synthesize some, have the requisite enzymes to synthesize the purines and pyrimidines. Those are just the base part of a nucleotide base. that can then be used to make DNA and RNA. So just know that the nucleotide bases that are needed for nucleic acid synthesis can be produced by enzymatic activity in these cells. Amphibolic pathways are ones that can operate in forward and reverse direction. We've just been talking about a couple of those. We've been talking about pathways that can, we just talked about producing Krebs cycle intermediates as by way of catabolizing the amino acids. Now we're looking in this picture at you making amino acids. Those are amphibolic pathways that can do both. And here's just a big picture look at a bunch of different pathways you've already talked about. Take your time and just look through that diagram. It's just showing what we've already talked about. Amino acids can be catabolized to produce chemicals that can feed into the energy scheme in glycolysis in the Krebs cycle. Right, here's an amino acid going in to produce energy. Or we can take those intermediate compounds from these processes, these pathways, and form the amino acids back out of them. Those are amphibolic pathways. We can take glycerol and oxidize it to make energy by feeding it into the glycolysis pathway and then down to acetyl-CoA, or we can take the intermediates, glycerol 3-phosphate, and convert it back to glycerol, and that's an amphibolic pathway. So you get the idea. So there's a whole bunch of reversible pathways that microorganisms have that they can either be oxidizing fuels for energy production, or they can be using these same pathways to produce building blocks that they need for anabolism, for building stuff. So that's our story for chapter 5, and we'll go on to a little bit less abstract and complex chapter 6 next, so join me for that.